So good afternoon, everybody. My name's Euan Wallace. I'm the chair of the Department of Obstetrics and Gynaecology, and it's my distinct privilege to welcome you to this open research event this afternoon, slightly differently this year for the very obvious reasons. My job is simply to welcome you. I'm going to introduce now Professor Stuart Hooper. Stuart is the Director of Research for the department and the Director of the Ritchie Centre. And then Stuart's going to introduce some of the discovery research opportunities for the department, and then introduce our colleagues, our research colleagues, who will share with you some of their research. Stuart, over to you.
Okay, thanks, Euan. Hopefully I can share my screen. Can everybody see that? Yes? Okay, for those of you who were just recently at the ONG previous meeting, you would have found out or heard about the Ritchie Centre, which basically sits across both the Department of Obstetrics and Gynaecology and the Hudson Institute. And during that session, they would have introduced to you the fact that the Ritchie Centre actually has a number of research themes, and which are women's health, fetal and neonatal health, infant and child health, infection, inflammation and immunity, and cell therapy and regenerative medicine. Now, my job today is actually to talk about the type of research that we do. And in a sense, what we have is a diverse range of expertise and projects at the Ritchie Centre, which really cover all of the different aspects of research. We do fundamental research in the laboratory. We do research with patients in the clinic. We do big data, epidemiological research, or research looking at outcomes. What type of models do we use? Well, we do our research in pregnant women and their infants. We use a variety of animal models, sheep and mice in particular. But we also go down deeply and look at the minutiae of what's happening in the cell system. So looking at the very complexity of what's happening down there. So fundamentally, what we do, what our vision is to work along all stages of the continuum between women's health, pregnancy and baby health. And it actually is not a one-way direction, because as you well know, that actually the baby can affect the mother. Indeed, in our women's health themes, we have research being conducted looking at endometriosis, and indeed looking at bioengineering solutions for pelvic organ prolapse, which of course gets damaged during childbirth. In the infant health area, we have research looking at how infants sleep, and particularly what happens with obstructive sleep apnea. We have projects going down that continuum, looking at pregnancy, trying to reduce stillbirth rates. How can we treat preeclampsia, which is a major killer of women, pregnant women? What's the effects of maternal obesity? Indeed, with what's happening in this current age, obesity is becoming a major problem. What's the impact of telehealth coming out of COVID-19 and the pandemic on maternal fetal medicine? And how does fetal growth restriction, how does it impact on the infant's cardiovascular health? We also have a lot of projects in stem cells, the stem cells from placenta or the umbilical cord. We're using bioengineering techniques to look at how do we prevent or cure diseases, and some of the diseases are stroke, chronic lung disease in preterm infants, as well as protecting the newborn brain. We also look at what happens at birth and preterm birth. What's the timing? How does the timing of umbilical cord clamping affect the infant? How do the lungs aerate at birth? And we can use the synchrotron to generate images like this, which we can turn into movies, as you can see in the screen here. We're looking at how treating neonatal seizures. We have clinical projects looking at NICU emergencies, how can we avoid them, and improving oxygenation and preventing preterm brain injury in infants that are in the intensive care unit. We also have a heap of projects looking at how we can support infants as they transition to newborn life. particularly preterm infants, growth-restricted infants, severely asphyxic infants, infants with a congenital diaphragmatic hernia, and infants delivered by caesarean section. So this is sort of just a very broad picture of the type of research that we do in the centre. And now I want to hand over to some of the supervisors who will actually talk a little bit more about the specifics of the type of research that they do. So I'll hand over to Graeme Polglase.
Thanks, everyone. I hope you can hear me and see the screen. Thanks, Stuart. My name's Graeme Polglase, and I'm head of the Fetal and Neonatal Health, Respiratory and Cardiovascular Research Group within the Department of Obstetrics and Gynaecology. So really our research focuses on a major issue that is a big problem worldwide, and that is of premature birth. In Australia, about 8% of all babies are born premature, and worldwide, being born premature is still the single greatest cause of neonatal morbidity and mortality. So what my team does is investigates how events that occur prior to or at the time of birth can lead to lung, heart, and brain injury. And things that can occur prior to birth include being born premature, of course, being born growth restricted, asphyxia, infection or inflammation, or things that can occur right at delivery of the newborn.
Sorry, Graeme, have you got slides there?
Oh, can you not see them?
No. Maybe sharing a different screen.
Am I sharing the wrong screen?
Must be.
We can just see you, Graeme.
There you go.
Is that me?
Oh, no, that's family.
That one?
Yes. Just in presenter mode, pop it in. Yep.
Okay. Sorry, everyone. I'll do it in presenter mode if you don't mind. Sorry, and at birth, we've got things that can occur at birth, such as delayed cord clamping, the requirement for respiratory support, requirement for oxygen, or the need for chest compressions or advanced cardiopulmonary resuscitation. Now, our research group consists of respiratory, cardiovascular, and neurological physiologists, as well as neonatologists, and this helps us to ensure rapid translation into clinical practice. So potential projects we have include targeting therapies that protect the fetal brainstem, which is the main cause or regulator of breathing, and whether they can reduce the need for respiratory support in preterm neonates, whether anti-inflammatory therapies reduce lung and brain injury resultant from long-term respiratory support, can we improve neurological outcomes of asphyxiated newborns requiring advanced cardiopulmonary resuscitation, improving the cardiovascular transition in compromised newborns. The first four are all in animal studies, and we've actually got some human-based studies looking at resuscitation videos of live deliveries from Monash newborn and trying to figure out whether we can assess the need for resuscitation based on general appearance of the human newborn during that initial resuscitation. Thanks.
Now, is my screen sharing?
Not yet, Caroline.
Right. Okay. Here we go. Is that right now?
Not yet.
Yeah, not yet. Oh,
We can see you, Caroline.
is that one working? Oh, I had this all set up. I don't understand why it's not working now. Oh, okay.
Yes, that's coming up now.
Right. Okay, good. Thank you. Okay, so my name is Caroline Gargett, and I look after the women's health theme. So I'm going to talk to you a little bit about some of the projects available in women's health. And we have a very big program of research on endometriosis. Now, endometriosis is a disease that's not much known about, even though it affects 10% of women. However, we've got a number of different projects in the discovery area, and particularly looking at what causes endometriosis, and projects looking at how the risk genes influence the cells of origin that we think might cause endometriosis, so endometrial stem progenitor cells. We've got some work in the area of diagnostics, looking at exosomes, and also perhaps using menstrual fluid as a biological diagnostic fluid. We have projects in personalised medicine using endometrial organoids. So these are patient-specific ways of growing the endometrium, and we know we can determine their genetic risk and then look at how they behave in vitro and eventually test drugs. And we have another collaborative project looking at a novel therapeutic target in mouse models of endometriosis. So in blue are the potential supervisors for these projects. We also, as mentioned by Stuart, we have a number of projects available in pregnancy disorders. This is not my area, but this is a very active area of research in the Ritchie Centre and the Department of Obstetrics and Gynaecology. So looking at things like maternal obesity on placental function, improving blood vessel function in women with preeclampsia, and looking at maternal nutrition and the effects of energy on energy homeostasis. And you can see here the supervisors that are available for projects if this is your interest. We also have a very big multidisciplinary project on pelvic organ prolapse, which, as Stuart said, comes from birth injury. We have projects trying to identify and then treat childbirth injury to prevent prolapse, which actually occurs much later in life. And as part of that, we've been developing endometrial mesenchymal stem cells as a cell-based therapy. So there's projects there at quite high in molecular, but also translational work. And then there's a bioengineering part where we've been doing 3D printing and nanofiber biomaterials and using cells to treat POP. And I'll just finish that POP affects 25% of all women and 19% need a surgery. So it's a huge problem that we're addressing here. Thank you. And stop sharing.
Hi guys, I believe it's my turn now. And given that no one's speaking, I'm just gonna go right on ahead. So I hope you can see my screen. Is that right? Somebody respond, tell me that you can see this.
Yes, we can see your screen.
Excellent. I will stop panicking now. So first off, thank you guys. I've just had a look at the number of participants. So there's quite a few students who've logged on for this virtual open day. So thanks you guys for showing up while the senior scientists are sort of scrambling with technology and trying to work out what's going on. So I'll just speak to you a little bit about one of the projects that we've got available this year. It combines two areas that are of interest in the Ritchie Centre in general, that's stem cell biology, and you've just heard Professor Gargett talk about this, and neonatal lung injury, which our colleagues earlier on had spoken about. So our approach in our lab is to use both animal models of injury, but as well as that we look to tissue culture and confocal microscopy for a fair bit of the work that we do. To give you a sense of this, our team works primarily on the amniotic epithelial cells. So they are one of a number of different perinatal stem and stem-like cells that you can get from gestational tissues. And you might recognize a few of these stem cells on the screen now from your undergraduate lectures. The cells that we work on are the ones that come from one of the two fetal membranes that surround a baby during pregnancy. And the top left-hand corner here just shows you the two fetal membranes, the amnion and the chorion, and we physically peel them apart, and you can see the forceps here in panel B showing that you can actually peel these two and separate them. And the panel of images down the bottom are just phase contrast images that we can take from a tissue culture type of microscope. And you can see how these cells kind of grow in culture. We spent a number of years understanding what these cells do and how they do what they do. And we're actually in clinical trials now to understand if they are well tolerated in patients across different disease types. What we've been most interested in of late is actually what the cells shed. So it appears from our research that involved honours students and PhD students that these cells don't actually need to engraft. So we used to think that stem cells engraft and turned into new tissues, and that was how they worked. And now we know that it's actually through other methods broadly referred to as paracrine signals. So one of the things that these cells do is they release these nanoparticles, and the panel on this right-hand side here are actually electron micrographs, so images taken from an electron microscope. And that cup-shaped type of particle that you're seeing, they're referred to as exosomes. So they actually develop within a cell, so they're called multivesicular bodies, and then they fuse to the side of the cell membrane and get released into the surrounding environment. And we think these are actually little particles, little packages that contain the good stuff that these cells release. And this is what we're looking into at the moment, how these exosomes actually communicate between the stem cell and the recipient cell. And the image that you see on the right-hand side here was actually captured by a previous honours student of ours. The blue dots that you see are actually cell nuclei, the endothelial cells. And as they branch to form this beautiful network of blood vessels, what we see is that the red dots, those are actually exosomes that were previously tagged. So what we know is that the accumulation of the exosomes at these branching points is part of how they do what they do. And what we're looking for is an honours student that would continue this work and take it towards a publication. Where this ties in with lung development is actually that in lung development, angiogenesis, so the development of new blood vessels, actually goes hand in hand with the development of new air sacs, so the development of alveoli. So this is just a real quick snapshot of the animal model that we use. So one of the earlier questions that we saw was what sort of techniques and what sort of models get used. So both sheep and mice, so at two ends of the spectrum that we use to study neonatal lung injury. The other method that we also use is actually 3D organoids. So I've just got a little picture in here that shows you that there are actually a number of different stem cells that exist within the lung, different hierarchy of stem progenitor cells in the lung. And you can actually sort them out and culture them in matrigel, for instance, which is a growth matrix, and actually watch them grow. So one of the questions that we're asking now is how exosomes can actually influence the growth of these 3D organoids and how they actually play a role in lung repair, because we can mimic lots of different types of lung injury in vitro. So thank you very much for your time, and I hope you guys have a great night.
Rob, you're still on mute, I think.
Okay, how's that?
Yeah, that's better, Rob. We can hear you now.
Thank goodness. Okay, I hope you can see my screen as well. So thanks very much for everyone being here. My name's Rob Galinsky. I head up the Developmental Neuroscience and Physiology research team, and our team is very closely integrated with the perinatal transition group and the neurodevelopment and neuroprotection group as well. So we really focus on understanding the mechanisms that contribute to preterm or brain injury around the perinatal period. And we're most interested in this critical window here, the last trimester of pregnancy. And the reason for this is because it's a very dynamic time of brain development. And you can see that through this picture here. You can see a very simple structure going to a very complex structure that has this very unique, distinctive folding pattern. And we know that the developing fetus can be faced with stressors during this critical time of brain development. And this can lead to things like lifelong disability and in some cases even death. So just a bit of context around what we do and how we do it. So the key problem we're focused on is what are the physiological and cellular pathways that lead to perinatal brain injury. We and others have identified that inflammation is a key trigger. And so with that information in mind, we're focusing on developing new therapeutic interventions that are targeting these key inflammatory pathways that get upregulated in babies that develop an injury in their brain around the time of birth. We're also working on improved injury detection. We know that the sooner you treat the injury, the better the outcome. And so we do this using a few methodologies. We study things like fetal behaviour. So we know that the fetus will breathe, it will move, and it will display distinct patterns of REM and non-REM sleep. And so we measure these things preclinically in order to try and gauge if we can detect alterations in these behaviours, to give us clues as to when we can, or the earliest possible time that we can detect a brain injury starting to evolve. We also use magnetic resonance imaging. This is the gold standard for detecting a brain injury around the time of birth. So it's capable of identifying changes in brain structure, but we still don't really understand what those structural changes mean at the cellular level. And so our team's working on improving our understanding of this relationship between changes in brain structure and changes in cellular pathology, so that we can improve how we use MRI to diagnose a brain injury in a newborn baby. And we're also working on developing more effective brain imaging techniques. Stuart's already given you a really beautiful picture, actually video, at the start of the presentation showing the lung getting aerated. And we're using similar technology together with Stuart and his colleagues in the physics department to see if we can improve the way that we image the brain. So what sort of technology or techniques do we use? So we use a very integrated approach. We use electrophysiology, so things like the electroencephalogram to study brain function. We also can measure fetal movements using electromyography, and we measure ECG, or fetal heart rate, as a marker of autonomic nervous system function, because we know that the autonomic nervous system is controlled by the brain. We measure cardiovascular physiology, so things like blood pressure and blood flow. These are important indicators of brain health. And we combine these physiological measurements with brain histopathology on postmortem sections. And we've got a couple of collaborations with teams in Switzerland and North America, where we are using or developing advanced MRI techniques for better detection of newborn brain injury. This is the last slide. This actually comes from a couple of my students. They call the lab that we work in Papa G's Pizzeria. Now, I'm Papa G, and the pizzeria comes from the fact that we have a combined passion for eating pizza. We're not doing that much together at the moment. And so I think what they're trying to say is that if you want to join the team, you also need to really enjoy pizza, but I'm not that biased. So please feel free to touch base. My email's there, and I'm happy to take questions during question time. Okay, let me unshare.
Hi everyone, my name's Suzie, and my little presentation to you guys today follows on from Rob's beautifully, because we do a lot of complementary work and we work together a lot. And really, I'm just going to give you a little overview of the neurodevelopment and neuroprotection research theme at the Ritchie Centre. In Australia, we know that about 10% of pregnancies are impacted by complications that may alter brain development. These can include fetal growth restriction, or FGR, intrauterine inflammation that Rob alluded to just previously, preterm birth, and perinatal asphyxia. Now, the techniques that we use to try and understand these span all of those things that Stuart talked about earlier. We have studies in which we examine placenta and other things in pregnant women and neonates. We have animal models of all of these 4 compromised situations in both particularly sheep, but also rats, and we have some cell-based work as well. And together we use all of these to work to reveal the cellular mechanisms that mediate fetal and neonatal brain injury so that we can test and implement targeted therapies to prevent long-term neurodevelopmental deficits, including cerebral palsy. I'm just going to give a really light overview of what these potential projects might look like. I encourage you to go and have a look in the booklet, but here's a little overview of what you might find from our group. One question that Emily Cam is asking, together with Kirsten Palmer, is whether maternal obesity alters placental function, and in turn, how does this mediate fetal well-being and fetal growth, particularly related to brain development? If you join Courtney McDonald and myself and other members of the team, you can look at examining stem cells from human umbilical cord blood to see whether or not they're neuroprotective. You could join Beth Allison and other members of the team to look at the impact of fetal growth restriction on the developing cardiovascular unit. So this really explores this intrauterine environment and how that impacts neonatal health. You can look at the long-term behavioural deficits associated with fetal growth restriction and neuroprotective therapies. And I lead this research on lambs, and we have a large research team that participates in this work, and it's really cool looking at behavioural profiles in lambs after they're born. Or you might choose to look at novel therapies to reduce neonatal seizures following acute hypoxia in birth. And again, this is using an animal model in lambs, and this is led by Tamara Yorno. That's all I'm going to talk about with respect to the work in our group. I encourage you to have a look in the booklet, but now I'm going to introduce Madeline Smith. Maddie is a PhD student in my group and in the Department of Obstetrics and Gynaecology, and was an honours student in the Ritchie Centre too.
Lovely. Thanks, Suzie, and hi everyone. I hope your uni semester's going well and you're all doing okay. I'm going to talk just a little bit about the student experience at the Ritchie Centre, and I thought I'd start off with some advice, and that's to find a project that excites you and you'll have an amazing experience at the Ritchie Centre. I think before you meet with potential supervisors, it's a good idea to do a little bit of background reading before you meet to understand the project and have any questions prepared so you can have a back and forth conversation. The Ritchie Centre is a very supportive community, and the community of staff and students really sets you up for success. I found that when I started honours, PhD students and staff were always happy to chat with honours students, and the community is just amazing. So the Ritchie Centre falls within the Hudson Institute of Medical Research, which has the Hudson Institute Student Society, or HIS. The HIS committee provides students with lots of social opportunities. And we can go to the next slide, including trivia nights, movie nights. There's a netball team, which was held pre-COVID times, where we had games once a week. There's also the 3-minute thesis competition, which is really fun, where PhD students present their research in 3 minutes in a creative and engaging way. And there's also monthly dumpling nights at Pings in Clayton. And there's a student retreat, which is held every second year. And in 2018, I think it was, we went to Phillip Island for the night. Students presented their work, and it was a great opportunity for staff and students to get to know each other outside of a work setting. We also have the MHTP Research Week Symposium, which is also a conference-like event where students have the opportunity to present their work. So there's heaps of social and career developing opportunities at the Ritchie Centre, and I've thoroughly enjoyed being a student there. So thank you.
Thanks heaps, Maddie.
Great, thanks everyone. So coming to the end of our time, sadly, with you, I hope you've got a sense of the very rich diversity of research opportunities that are available in the Department of O&G our partner institute, the Hudson, through the Ritchie Centre. Whether you're interested at a cell or subcellular level, whether you're interested in using animal models of human disease, all the way through to clinical applications. I think what you get a sense of is that our research in the department is very focused on trying to address the most important health and disease burden outcomes for women and their children. We'd love for you to join us. I've been looking at lots of your questions in the Q&A line. Because of the diversity, really irrespective of your major undergraduate streams, there are opportunities for you in women's and perinatal health. So Stuart, is there anything else you wish to say from your side?
No, thanks, Euan. Just other than, as I think Suzie rightly said, just have a look at the booklet, but look at the sort of area that you want to work in, and then go and see the supervisor and talk to them, because quite often there are other projects other than those ones that are listed in the booklet that you might be able to do. Okay, so they're only a sort of a list of just a small list of the projects. There's lots of other projects that may arise as well. So I implore you to go along and talk to each of the supervisors.
Wonderful. Well, thanks everyone. Hope you found something of interest, and hopefully we'll see you next year.